All right, so let's go ahead and get started. This is where we ended with phases of gastric secretion, and I believe this picture, right? Excuse me. So remember there are three phases. You have your cephalic phase, your gastric phase, and then your intestinal phase. So this is, again, one of those summary slides you'll come back to. But this is the explanation, the explanation parts. So, like I said, it's Friday. That was Wednesday. 2 days ago, thank you. Smelling my coffee, right, automatically gets the cephalic phase of gastric secretion going. Okay? And don't, it says cephalic. That doesn't just mean brain, because you see that some of the stimuli are mechanoreceptors. Where are mechanoreceptors located? In the head. Mouth, pharynx, esophagus. All right, so mechanoreceptors, so food coming in, that's still part of the cephalic phase. As well as chemoreceptors, that's our smelling, that's our tasting of food. So that says food's already in our mouth as well. Just thinking about food. Have you guys ever, I'll go back to my go-to, like pot roast. And you start thinking about pot roast and you like feel like tingling in your parotid glands. You said, yeah, you felt it. I usually don't see nods like that. She's like, You bringing some in next week? And automatically your salivary glands, like you can feel that in your mouth is watering and you're like, oh, God, yes. And then find out that you're not getting any. So that can stimulate, and that's about 30% of your total acid response. So that's like less than half. So it's preparing. Most of your acid response comes from the gastric phase. Yes. I can't hear you.
Oh, do you have a microphone?
You can't hear me?
Oh, that's why I moved up here.
Can I talk louder? Is that good? All right. I do have a microphone. Yeah, he does speak. I do. I don't like to use it. So I'll just talk louder. I'm going to be tired, so tired. Okay, so gastric phase, where does that occur? Stomach. Because the other word for gastric is stomach. So that's where so cephalic phase is strictly directed by the nervous system. When food enters the stomach, other factors come in play. So like different sensors, an increased pH stimulates acid release. Distension of the stomach, so mechanoreceptors, nervous system, peptides and amino acids. Once you start breaking down proteins into their individual constituents, amino acids, they bind to receptors and say, hey, let's secrete some more acid because we got some shit here to break down. And these will also stimulate acid response in the stomach as well. Which is why drinking alcohol and drinking coffee are said to cause ulcers because they increase the amount of acidity. All right, the last phase is the intestinal phase. All right, so you've gotten from the stomach, you're moved into the duodenum of the small intestine. If the small intestine is receiving contents, it's saying, hey, I'm getting food. That means I need to tell the stomach, hey, I'm getting food, so you need to turn on some more acid secretion. All right, notice that only counts for about 10% because all this other acid secreting stimuli have already had their effect. So it's the intestine saying, hey, just a little bit more. Come on, push. Like you get, I mean, some of you in here are runners. Like you get to like a marathon and you're at 23-mile mark and you're like, oh my fucking God, I can't possibly finish. You got to do that last 10%. I'll tell you my marathon story later. Also amino acids, because it's in the duodenum where you really start breaking everything down into the smallest possible parts to get absorbed. So amino acids will also stimulate acid secretion, because acid activates what? You know, I have a little app on my phone that has crickets chirping. What does acid activate in the stomach?
Oh,
Oh, I needed to tell you where.
I can't think of his name, but you cut off the thing.
You cut off a thing and it pepsinogen converts it to pepsin. And what does pepsin do? Break down proteins. Okay? So, the intestine is saying, hey, amino acids are coming here. That means you're giving me proteins. You need to make sure that all of your pepsin is activated. This is, I love writing on my PowerPoint. All right, so this is just showing you the acid response to a meal. What I want you to notice about this graph is that, you know, here's your basal acid secretion, and then as soon as the meal is in front of you and you start eating, what happens to, the blue line represents pH. The green line represents the volume in stomach. So what that says is when you're taking food in, what happens to the pH of the stomach? It goes up, which is one of the stimuli for acid secretion. All right, and it's like a plop. It goes right into your stomach. So that's why the volume increases like precipitously. But acid starts initially precipitously. That would be the cephalic phase. And then it kind of gets slower and slower and slower when you are taking into consideration the release of gastrin for the secretion of acid and the release of other factors that will prolong the acid response. Okay? And then it peaks at a certain point, and then as your volume and the pH starts decreasing again, of course the rate of acid secretion goes down too, right? That makes sense because you don't want acid to be continuously secreted if there's nothing in the stomach. All right, so that's what this graph is showing you. All right, and this is basically saying the same thing in words. Early in the meal, acid secretion because of your cephalic phase. That is strictly nervous system. pH rises, and then you, like somatostatin keeps gastrin in check. As soon as pH rises, somatostatin stops, gastrin can start to be released, and then acid comes in. Okay? Late in the meal, you still have acid secretion, but it's decreasing because you want to start inhibiting the acid. You want to start, it needs to be tightly regulated, right? So you need to have a mechanism to bring it back down to baseline levels as the pH falls. So it's a really highly regulated mechanism. Early in the meal, mostly late in the meal, though. All right, so this is just a summary slide of the stomach regions involved. Actually, not the stomach regions. The GI regions involved. The stimulus, all right, so the stimulus would be acid less than 3, releases somatostatin to inhibit gastrin and inhibits acid secretion. So that's how you're reading this chart. In this area, where's the oxyntic gland area? Where's the oxyntic region of the stomach? Is that down here or is that up here? What? No. That's the pyloric region. Oxyntic region is the top. Another name for the oxyntic region? Pardon? Fundus body. Another name? Well, cardiac regions there too. What's the other name that encompasses all of that? Begins with an O. Rhymes with SNORAD. The ORAD region. Okay, in the duodenum, acid getting into the duodenum, hyperosmotic solutions cause secretin release, nervous system reflex. Some other shit goes on. inhibits gastrin, secretin inhibits gastrin, also directly inhibits acid secretion, and so on. So this is a summary of how we shut off that acid response, in other words. All right, and if you wonder what GLP is, this is why I gave you this sheet, right? Go down here, look at GLP, glucose-dependent insuliноtropic peptide. That's what GLP is. And you notice it inhibits gastric acid secretion. That's why I gave you this. All right, so now we've had a meal. We've secreted acid. We've secreted all these factors to promote the secretion of acid. We've got pepsin now in there because acids activated all our pepsinogen. So now we start mixing all this together. And how many types of muscle fibers, how many directional muscle fibers do we have in our stomach? 3. Circular, longitudinal, and oblique. Right? So we're mixing all of that around, churning it up until we make this paste called chyme. And I think I mentioned that on Wednesday. Once it's in the stomach and all mixed up, it's chyme. Because you don't want that Oreo trying to fit through the pyloric gland, or the pyloric region, the pyloric sphincter. All right, so it all needs to be in a fine paste so that all the enzymes that are required act on the individual constituents. Right, so this is basically the end of mechanical digestion. It's just broken everything apart. The acid, the pepsin, it's broken everything apart and made it a paste. All right? And then it starts pushing up against the pyloric sphincter. If it's pushing up against the pyloric sphincter and the pyloric sphincter is closed, what's going to happen to this region? It's going to stretch. What do you think that's going to do to the pyloric sphincter? Open it. Okay? So that's one of the signals to open it. All right. But we have to regulate very carefully how much chyme enters the duodenum, because the enzymes that are in the duodenum operate at a pH of 7. What's the approximate pH of food coming from the stomach? 2-ish, 3-ish. All right, so we need it to go into the duodenum, raise that pH up to neutral really quickly so these enzymes can function. So it's got to be regulated very properly, it's got to be pretty slow. All right. We also have to slow it down enough for nutrient absorption, because what happens, we've all probably done this. I know I've done it too many times. You drink just a lot of water really quick. Half hour to an hour later, what are you doing? Peeing out your anus, right? Because it all just rushed through. Okay, you don't want that to happen because you want things to be absorbed. None of that water got absorbed because you literally peed out your butt. I'm just being real. I remember being a kid and boys getting together and arguing about how many holes a girl had and whether or not they pee out their butt. Yeah. No, I'm teaching. I'm here to tell you that doesn't happen. But that's what this is like. So you want things to go slow so enzymes can work, 1, and 2, absorption can occur efficiently. All right, so there are some factors that favor emptying of the stomach, and there are some factors that inhibit the emptying of stomach. So these are factors that favor. That's why you have a green belly over there, green stomach. All right, be careful. Now that you guys are, in PA school and you're going to be PAs, this is not your stomach. All right? I know we want a flat stomach, but our stomach is this little organ that's over here, not this. Okay? So, increased tone of ORAD stomach. What do we mean by increased tone? What's it doing? Contracting. Not stretching, contracting. All right, so it's contracting, increasing pressure. Forceful peristaltic contractions. That's what these are showing right here. So you start moving things along. Decreased tone of the pylorus, so that means decreased tone would be it's relaxing. Increased tone contracting, decreased tone relaxing. And then absence of segmental contractions in the intestine, so you don't have peristalsis going into the intestine. All right, those are what favor stomach emptying. All right, but why did that move? There's the arrows. Let me just get that over with. All right, factors that decrease gastric emptying, just the opposite. The ORAD region of the stomach relaxing, right? So it's not pushing, so that's going to cause things to back up a little bit. Decreased force of peristaltic contractions. You notice that the lumen of the stomach is not as small because these contractions aren't as big. Increased tone of the pyloric sphincter, so it clamps down and says, whoop, nope, you got to stop. And then segmental contractions in the intestine. So that's the intestine saying, all right, I've got what I needed. I'm going to start moving things along because we got to prepare along because we have to prepare for the next load. Okay? So this is a constant back and forth thing after a meal. Favoring emptying versus inhibiting emptying. All right, and one of those reflexes that are involved in this is something called the enterogastric reflex. And I mentioned this on Wednesday when I talked about vagal reflexes. This is a vagal reflex. All right, so what happens is, follow the numbers. You're pushing food, or chyme rather, from the pyloric region into the duodenum. What's happening to the duodenum? It's stretching. It has mechanoreceptors in there. That stretching of the mechanoreceptors sends a signal to the central nervous system via the vagus nerve. Where in the central nervous system is this going to? Do you remember? We talked about it. Where are your digestive reflexes? Begins with M, rhymes with schmenula. Medulla. Thank you. All right. Then the medulla says, hey, all right, if this is a reflex, do you have to think about it? No, it's entirely underneath conscious control. So then the medulla is going to send signals back to the stomach. Notice that the signal starts in the small intestine and then ends in the stomach. And the stomach is going to relax. And it's going to relax because the medulla is saying, hey, you're pushing too much into the duodenum. Let's slow down our parasympathetic impulses. And if you slow down your parasympathetic impulses, the stomach starts to go. And that's that relaxation of the ORAD region that we talked about, inhibiting gastric emptying. All right, so the enterogastric reflex, the purpose is to slow down gastric emptying. And why do we want to slow down gastric emptying?
Good factors.
Pardon?
Good acid level. Like pH in the small intestine for the enzyme working.
pH, enzymes, and absorption. Good. All right. So, there are also other factors within the intestine. So, by intestine, we're specifically talking about the duodenum here. That will decrease gastric emptying. So, they're going to be in the mucosa, and they're going to be stimulated by osmolarity. What does osmolarity mean?
Exchange water.
Okay, that's what osmosis is.
Yeah.
But what's osmolarity? I say something that's a high osmolarity or a low osmolarity?
Like
Concentration of solutes. Good. So that's the concentration of solutes. So if it's like pure water or like salt water, that's going to stimulate these receptors. Acid, of course, we already saw that. Fats, lipids, and protein. are receptors that will help decrease gastric emptying. All right? And they do so, by triggering the enterogastric reflex. Okay, so it's not just stretching. It's also these other things. So fats and proteins. CCK. Take out your paper. What's CCK? Cholecystokinin. Break that word down. Chola refers to? Pardon? Okay, yeah. Cysto, bladder, gall, or cholesterol, bladder, gallbladder. Kinin, kinetic energy, means movement, right? Kinin. Anytime you see kinin, that usually means like go or activate or something like that. So, cholecystokinin, a long explanation for 3 small letters. That increases gastric distensibility, which decreases gastric emptying. Okay, so CCK, upon fats and proteins entering. So when fats and proteins enter the duodenum, they bind to receptors and cause the release of CCK. CCK is a hormone. That means it goes in the blood and comes back to the stomach. All right, decreases gastric emptying because you want to be able to digest those fats and proteins. So one of the jobs of CCKs is to facilitate the digestion of fats and proteins. And this is one of the mechanisms. Cholecystokinin actually has 3 major functions we'll talk about. Acid. Acid into the small intestine triggers a decrease in gastric emptying by a direct intrinsic neural reflex. So where is this reflex occurring? If it's an intrinsic neural reflex. Pardon? Did you say that?
Pons.
I didn't hear you.
Pons.
No, that's not intrinsic. Intrinsic to the digestive system. Pardon?
Would it happen right there at the site?
So what's that called?
Enteric nervous.
The enteric nervous system, right? And then involvement of other hormones. Forget that last one. I don't even know why I put that in there. All right. So now we want to go back and talk about protection. We've talked about, the stomach and all this acids getting secreted, and then you have pepsin and then we're emptying and preventing emptying. So how do we prevent the stomach from I feel like I'm missing a slide. How do we prevent the stomach from eating itself or getting digested? Well, there's 3 general mechanisms. 1 anatomic, 2 physiological. The anatomic is you have in your stomach, the mucosal cells, but they have tight junctions between the mucosal cells. And what do tight junctions do? They provide a solid protein barrier between the cells so nothing passes between them. So think of it as like stitching between the cells. All right, that's anatomical. Physiological, if a hydrogen ion happens to go into a mucosal cell, so it will diffuse directly into a mucosal cell, the mucosal cell will spit it right back out, pump it right back out into the lumen. All right, as a mechanism to help defend itself. All right, and so that's what this means. And then we have mucus secretion. And usually when we think about protection of the stomach, the first thing you think about is what? Mucus. All right? So, things that strengthen this barrier: mucus, bicarbonate secretion. What's bicarbonate and why is it important? For protecting against, right, it buffers acid. It buffers acid. It's a base, so bases buffer acid. Gastrin, prostaglandins. Prostaglandins are vasoactive molecules. And epidermal growth factor. So all these things strengthen. If you have all of these intact, you have a healthy barrier, you're less likely to get ulcers. all right? Because ulcers are ultimately caused by the end product is too much acid destroying cells. Things that weaken the mucosal barrier, H. pylori, that's an infection within your stomach, and usually they say this is the cause of stomach ulcers, but every single one of us probably has H. pylori in us right now. I don't know if you've heard that before. People like to say, oh yeah, people get this infection. Everybody has it. Everybody has it. But we don't all have ulcers, right? So it's not just H. pylori. Aspirin and NSAIDs directly have an effect on decreasing mucus secretion. So that's why when you have patients that need pain control, you want to be wary about giving them NSAIDs or giving them aspirin, because that's going to weaken their gastric barrier, and you could end up with ulcers as a result. Ethanol, similar effect to aspirin NSAIDs. In fact, if you go out to Canvas, I have a video that it's kind of a neat video that shows specifically how aspirin and NSAIDs decrease mucus secretion. All right, bile salts as well, also weaken. You have these things, you're going to likely get a damaged barrier, more prone to ulcers and gastritis. All right? Now, what's the mechanism behind how acid destroys the mucosal barrier? All right, when we talk about the mucosal barrier, we're talking about the cells, not the mucus, the cells. All right? What makes acid? If I say, here's an acid, what makes it an acid? Hydrogen ions. All right? So when we talk about acids, we're talking about hydrogen ions. If you create a situation where hydrogen ions, remember I said one of the protective mechanisms is if hydrogen ions get into the cells pump them back out. But what if you overwhelm those cells with hydrogen ions and they can't keep up with the pumping out? What's going to happen to the pH of those cells? What happens to proteins in cells with a low pH? They denature. What happens to denatured proteins? Do they work anymore? No. So if they don't work anymore, you can't maintain the cell integrity and the cell dies. Okay? So that's how decreased cell pH would lead to cell death. All right? And then you have a vicious feedback cycle where you have cells dying. You start leaking histamine, which produces more acid, also brings more blood to that area. More leakage of hydrogen ions, more cell death, and it's just a vicious feedback cycle. Okay? So once you overwhelm, and the key here is overwhelming the cells with hydrogen ions, that will lead to a peptic ulcer. All right? Does that happen like that? No, this takes a long time to occur, generally speaking. All right? So you generally have two types. Most of your ulcers you're going to see in the pyloric region or the duodenum. One of those two places. Why those two places? Why not in a fundus? Right. Gravity. Gravity. The pyloric region is the lowermost region, so gravity is going to make sure all the liquid stays down there. So you have gastric ulcers and duodenal ulcers. With gastric ulcers, remember that's in your stomach, pyloric region, the main problem is you're losing mucus. The stomach has a lot of mucus and a lot of protective factors. You start losing those protective factors, that's going to lead to gastric ulcers. Duodenal ulcers, on the other hand, their main problem is increased delivery of acid. You do not have as much mucus in your duodenum as you do in your stomach. So the protection of the duodenum depends on raising pH, right? Remember we want the pH to go up to 7 for those enzymes to work properly. So the main problem is increased acid delivery to the duodenum. So aside from increasing pH, and aside from more time for digestive enzymes to act, and what was the third one? More time for absorption. A fourth reason why you want to control gastric emptying is to prevent ulcers. All right. Now we're out of the stomach. And we're getting into the small intestine. All right. So the small intestine has three parts, as you know from anatomy: the duodenum, the jejunum, and the ileum. Like, Jesus. All right. Don't make this mistake. Notice the where is it? Spelling of the ileum. How do you spell ileum when it's a small intestine? I-L-E-U-M. How do you spell ilium when it's a bone? I-L-I-U-M. All right? Don't get those mixed up. Two different things. But they said, anyway, I don't know why they do stuff like that. I do, but it's not worth the discussion. All right, so three sections. We've already talked about the duodenum, and that's where we're going to focus most of the first part of this, is talking about the duodenum and its function. What time did we started about 9:30, right? I'm thinking 9. All right. Okay, so back up. 3 parts. The duodenum. The function of the duodenum is to finish chemical digestion. All right, so it's going to work with the gallbladder, it's going to work with the liver, and it's going to work with the pancreas to help finish digestion of everything that's getting pumped from the stomach into the duodenum. The jejunum and the ileum, their primary functions, absorption. All right, so the jejunum and ileum, absorption. All right, this, I think, is amazing. All right. What? So, if I were to spill water on this floor, how much of that water would get absorbed? Very little, almost none, right? What if I made the floor wavy? Would more get absorbed? A little bit. If you look at the floor, like you put cracks in it, a little more gets through. All right, do that. I have to make my analogies work. All right. If instead of this floor with the waves, I add carpet, how much can I absorb? A lot. All right. Well, what if I decide to make it like high shag carpet? We're absorbing even more. All right. So I've taken something that has almost no absorptive capacity and increased it 1,000-fold by adding folds of Kirkring. All right, so that's the folding. Villi. And then at the top of villi, I have microvilli. All right, so all of this folding, anytime you see folding in a biological sense, automatically think increased surface area. All right, so that allows us to absorb a lot. If a tennis court is about 200 meters squared, the absorptive capacity of our intestines is that to 2 times that. That's how much we can absorb. To me, that's amazing. And that's also not just amazing, but efficient, right? We just absorb the shit out of everything. That actually wasn't intended, but it worked. All right. Within the villi, we have a very specific structure. All right, and we've already talked about this structure when we talked about that the amount of oxygen that actually gets delivered to villi. If you remember that, and only 10% actually gets 20% actually gets delivered. 80% bypasses because the villi just need enough to keep them alive. Most of the capillaries are not designed to exchange oxygen and carbon dioxide. They're designed to take in amino acids and monosaccharides and things like that. All right. So, extensive capillary network underneath the villi. And then in each villi you have this brush border of microvilli, again, helping to absorb nutrients. All right. Within this capillary network, you have a central lacteal. And that lacteal is specifically designed to absorb fats, dump it into our lymphatic system, and then return it to circulation via our lymphatic pathways. All right. So, we've already talked about this in anatomy. Where does all the blood from the intestines go to first? The liver. Everything goes to the liver first because it has to be processed. But lacteals with fats don't go to the liver. They get dumped to the lymphatics. And where do the lymphatics dump into? Specifically, most of your lymphatics, remember that book from that picture from Bare Bones?
No.
Lymphatics dump into
Lymph nodes.
no, right about here. No, thoracic duct, and where does that empty into? Subclavian vein, right? So directly back to circulation. It's not that early in the morning. Oh, okay. All right. It's Friday. And you have an exam. All right. So, keep in mind, we're talking about the intestines right here and their absorptive capacity, but the intestines aren't just the only places where things get absorbed. In the stomach, alcohol gets absorbed, which is why you get drunk really quickly on an empty stomach, which is why they say eat food if you want to drink a few beers and be able to last the whole night. NSAIDs get absorbed in the stomach, and aspirin. All right, small amount of water. Duodenum and jejunum, almost everything else. All right, so the duodenum and jejunum, those are our absorbing carpets. The ileum will absorb bile salts. They get recycled back to the liver. So all the bile salts that are released by the gallbladder go into the ileum, and then they get recycled and put back into the liver to go back to the gallbladder. All right, and vitamin B12. All right. What factor and where is it secreted helps with vitamin B12 absorption in the ileum? Factor? Intrinsic factor. Secreted by cells in the... Begins with S, rhymes with stomach. All right. You guys are smart. That wasn't an insult, I promise. The colon absorbs water and electrolytes, and we're going to see why that is really important. So our main water absorber is the colon. And then the rectum can also absorb water, but also drugs as well, which is why you give suppositories. I remember when I first started working at Arrowhead as a nurse, and then I saw that there were some things were suppositories, and I'm like, we don't actually have to do that. We don't have to we don't have to do that, do we? And they're like, no, they usually do it because they're they're healthy people. They just have a drug problem, right? So they can stick something up their own rectum. Because it's one thing, like, all right, you have a patient that's laying there and they're in the ICU and they're not conscious, and all right, you got to put it up there. But like, Nick, sorry, dude, that would be weird, right? So, anyway, all right, when we absorb things in the small intestine, we have to cross 8 barriers, and we don't always think about this. So those 8 barriers, we have a mucus layer at the top, and then over every single cell in our body, we have this sugar network that kind of acts as a barrier. It makes our cells slippery, called the glycocalyx. Then we enter into the apical cell membrane, and in case you forgot, the apical cell is always facing towards the lumen when we're talking about, a barrier. So the apical surface, then it goes through the cell. Then it comes out the basolateral surface, basolateral, crosses the basement membrane, which is a bunch of proteins and carbohydrates, then goes to the capillary, has to cross the cell membrane of the capillary into the endothelial cell, and then into the actual capillary to be transported in the blood. That's a lot, right? That's a lot. And that's going on all the time. So when we talk about absorption, we're not just talking about, hey, we're in the blood. We're talking about a process of going through cells and getting into the blood. All right, so it is pretty extensive. Just be aware of that. I'm not going to ask any detailed questions about that.
We don't need to know the orders of the layers.
Do what?
Do we need to know the order of the layers?
Yeah, you should know the order of the layers.
Just making sure. The way you said that.
Hmm?
I was just making sure the way you said that.
Okay. All right, going back to anatomy a little bit. The intestines are suspended in the peritoneal cavity by peritoneal membranes. We have the mesentery, which holds all the folds together, and there's a picture of the that membrane and the blood vessels and nerves that are within there. Okay, and then we have the greater omentum, which drapes over all of the intestines and provides kind of a protective function. Most by protective, I mean not like somebody hits you in the stomach, somebody hits you in the belly, and you have a bigger omentum and like, oh yeah, hit me in the belly. My omentum, no, it's not that kind of protection. If you get an infection, the omentum will wall off that area and prevent the infection from spreading. All right. Now we move on to the pancreas, because the pancreas is directly connected to the duodenum of the small intestine. So as we get chyme from the stomach into the duodenum, we have to neutralize the acid, right? We already talked about that. And we have to make sure that all the nutrients, the macromolecules, the proteins, the polysaccharides, and the triglycerides get broken down. All right? So the pancreas allows us to do this. So what is the pancreas? This little organ right here that has a head, neck, body, tail. All right? We just talked about this in anatomy. All right? And the head is nestled into this C-shaped loop, the second part of the duodenum. All right? So it's nestled in there, and it's got a pancreatic duct that feeds from all of the acinar cells. All right? Remember, the pancreas has 2 types of cells, or two functions. It's endocrine, right? So it secretes insulin and glucagon and somatostatin. We won't talk about that here. It also has an exocrine function, and the exocrine function secretes substances into tubes, which would be this pancreatic duct, and then that empties directly into the duodenum. All right, through 2 sources. right? So everything that's being secreted by these pancreatic acinar cells flowing in the ducts, we call that pancreatic juice. Okay, pancreatic juice. And again, that's going to flow. Two things I want to mention here. You have two ducts. The major one is this hepatopancreatic duct right here, opening into the ampulla of Vater. You guys remember finding that in anatomy. And surrounding that, and it joins the bile duct from the liver, the common bile duct, and it's surrounded by a sphincter. So that sphincter keeps things closed. When you want to secrete pancreatic juices, that sphincter will open and allow stuff to drain into the duodenum. Okay? So I just want to mention that really quickly. And there's the sphincter right there. That's smooth muscle. Bless you.
Finger on both the ducts, or because there's the minor one as well.
No, the minor one, if it does have a sphincter, it's of little consequence. That the minor duct, the minor duodenal papilla, if you notice, it's more backflow that would allow that to come out. So whatever doesn't, if you have a lot of juices being secreted. All right. So what's in pancreatic juice essentially? All right. We have proteolytic enzymes, which means they break down proteins. These are also secreted as zymogens, right? Inactive forms. Why do we want them stored and secreted as zymogens in the pancreas? So we don't break down our pancreas. Good. All right, and one more. And these cleave proteins to peptides. So I have a big protein. It'll break it down into smaller peptides. Carboxypeptidase, which is secreted as procarboxypeptidase, gets converted to carboxypeptidase. That'll take these peptides and break them down into individual amino acids. Because ultimately, again, what do we absorb? We absorb amino acids. We absorb monosaccharides. All right, so we need to have them broken down into that. So these are the enzymes that break down the proteins. All right, for carbohydrates, we have pancreatic amylase. This is very similar to the amylase that's secreted by your salivary glands. Breaks down starches and glycogens to disaccharides. All right, to disaccharides. Notice it's not monosaccharides. That occurs later, although you can absorb disaccharides as well. All right, and then for fat digestion, the enzymes. We have pancreatic lipase, breaks down triglycerides into fatty acids plus monoglycerides. So by fat, we mean triglycerides. It breaks down phospholipids into individual fatty acids. Excuse me. And it converts cholesterol esters into fatty acids. All right, so pancreatic lipase, phospholipase, and cholesterol esterase are going to help break down the fats, different types of fats. All right, like I said, all the proteolytic enzymes are secreted as zymogens. Trypsinogen, chymotrypsinogen, carboxypeptidase converted to trypsin, chymotrypsin, and carboxypeptidase. So, I mean, literally you can, like the enzymes cut them, you can cut parts of the word off to make them active. I think it's fun. All right, I've already started making my lectures for next year. When I go over this, I'm going to change one term on here 2 times. We secrete all of these proteolytic enzymes as zymogens, but they got to get converted to the active enzyme, right? They don't get converted until they actually enter the duodenum. And there are enzymes on the mucosal cells of the duodenum called peptidokinase. No. Sorry. Enteropeptidase. Enteropeptidase. There's history on this. You guys remember molecular cell biology, right? What does a kinase do? It activates things by doing what? Putting a phosphate on things, right? But if you're looking at this, enterokinase puts a phosphate on trypsinogen and makes it trypsin, but we said trypsin has to be cut, and it does have to be cut. So kinase doesn't describe that function, right? So after they figured that out, they started calling it enteropeptidase instead, because that makes more sense, because it cleaves trypsinogen into trypsin, but kinase is still used commonly. So I prefer the more direct word, but whatever you guys want to use. So you can replace enteropeptidase here and here. So anyway, trypsinogen goes into the duodenum. This enteropeptidase converts trypsinogen to trypsin. And then once trypsin is made, the reason why you have this arrow going backwards is that trypsin can cleave off peptides to activate more trypsinogen to become trypsin. Does that make sense? So enterokinase gets a few of the trypsinogens converted, then trypsin converts the rest of the trypsinogen by doing the same thing. And that's what we call autocatalytic. Trypsin activates itself. But then it's also going to activate these other ones as well. So it's trypsin that gets first activated, and then trypsin activates all the other proteases. All right. Now, besides being secreted as stored and secreted as zymogens, within the acinar cells of the pancreas, we also have a trypsin inhibitor. There's a trypsin inhibitor within the pancreatic acinar cells that actively prevents the formation of trypsin. All right, so that's going to be very important. So you have double layer of protection: inactive enzyme and an enzyme that prevents the activation as well. All right. Usually, when we have cases of acute pancreatitis, it's because this trypsin inhibitor has gone off. Right? It's not working properly. Or not enough of it's present. Okay? I think it's the next slide that shows this. This trypsin inhibitor is also found in the lungs because you have a lot of enzymes in your lungs that are geared towards destroying, that are in the Roombas and neutrophils that are there, that will destroy things, but they will also destroy elastic tissue in the lungs. So we have this antitrypsin in the lungs to prevent destruction of lung tissue, so that those enzymes don't get overactive and destroy lung tissue and just work on the bacteria. All right. The other thing that pancreas secretes is bicarbonate. All right, and the stimulation to secrete bicarbonate comes from a hormone called secretin. All right, and that's another one that's on your sheet here. So secretin stimulates bicarbonate secretion. Bicarbonate goes into the duodenum of the small intestine and neutralizes the hydrochloric acid. All right, so those are the players. When do they get secreted? How do we regulate it? All right, bicarbonate is dependent on just acid entering the duodenum. Acid enters the duodenum, the pancreas gets a signal that says, all right, need to secrete some base. Put it in there. And that's going to happen all the time, right? Because you're always going to have acid coming from the stomach, no matter what. That's what comes from the stomach. The secretion of enzymes, however, is dependent on specifically what is entering the duodenum. If you have a lot of fats and proteins entering the duodenum, then you're going to secrete enzymes, the enzymes. So a lot of fat and protein entering the duodenum, you're going to secrete enzymes. All right, and those are regulated by the central nervous or the nervous system through acetylcholine, vagal reflexes, as well as cholecystokinin. So this is the second function of cholecystokinin. When you have fat and protein entering the duodenum, fat and proteins bind to receptors, cause the release of CCK into the bloodstream, which then goes through circulation back to the pancreas and causes the secretion of enzymes. All right? What was the other function that we said CCK had? So this is the second one. Pardon? Prevents gastric emptying. Right. All right. This is just a picture showing you the same thing. Reviewing everything we just talked about, so I'm not going to add anything new to that one. All right. Like the phases of gastric secretion, we also have phases of pancreatic secretion. Like, seriously? The good news is it's the same thing. You have the cephalic phase, you have the gastric phase, and you have the intestinal phase. What was the most important phase for gastric secretion? Or gastric phase. So what's going to be the most important phase for pancreatic secretion? The intestinal phase. Right? If you remember that, you're good. All right. Once we get to the intestinal phase, if we have acid, that causes this release of secretin, which will then tell the pancreas to release bicarbonate and water. Question: if you just tell the pancreas to release bicarbonate, why is that a problem? If you just tell secretin tells the pancreas to release bicarbonate and water. If you just tell the pancreas to release bicarbonate, what's the problem with that? Yeah, you need a liquid to be able to transport that bicarbonate. Okay, so that's what the water is for. I always, maybe I'll talk, I think I talk about it in a reproductive lecture, but it always reminds me of like sperm. Like if sperm were to just come from the testes without all of the fluid from the prostate gland and the seminal vesicles and the bulbourethral glands, would it just come out as like dandelions? All right. If you have fat and protein entering the duodenum, that causes the release of CCK, which then causes the release of enzymes. All right, we talked about what the enzymes are. If you have acid, fat, and protein, that will stimulate the vagal reflex and also help secrete enzymes as well. All right, so you have a protein mechanism and you have a nervous system mechanism. Right, and what do we know about protein versus endocrine versus nervous system? Nervous system is fast, but the endocrine system lasts longer. You guys want to take a break? Yeah? Okay.
Yours is like right out to the right. Girls' bathroom?
Girls' bathroom is right next to yours, too. You can either go down the hallway to the right or that way.
How long for
ask a question. I know the answer, but like the way that's interpreted in my brain isn't like able to be articulated. Like, so. You're like, what? I'm like, well, no, that's not makes sense in my brain, but like.
Is that why you guys don't say anything?
I don't know those things sometimes. Like, I know how like the function works, but then to like actually explain it,
Right.
which like you think would be the same thing, but for some reason my head is
No,
not.
I get it.
I agree with you on the whole. Like, I prefer anatomy over physio, but the nice thing about physio is, on the whole, it's not just blind memorizing. It's concepts and like you can kind of think through it. And
Right.
it's not just beating it into your brain, which I enjoy that. But
Right. So, why is the duodenum organized the way it is, and why is the pancreas right there? And that anatomically, okay,
they fit together.
but physiologically, explain. Yeah, why? Whoa. Before I. Well, this is why I also run things by you guys and talk to you and get reactions and feelings, and then you guys will come to me like Monday, or you'll email me over the weekend and be like, hey, maybe that's not so bad after all.
I would
Like, gut reaction sucks.
Could we get like a week's heads up so I can like really prepare?
Heads up. Now I'm nervous.
I'll make the decision by Monday.
And then we can quiz on Thursday.
I will make a decision by Monday and let you know all of the details.
Those coloring things on that quiz was so hard yesterday. The one that you have your practice on, is that what you're talking about?
I figured it out.
Oh, you did?
Yes. All right. So, obviously, we realized that, and maybe you guys in the back weren't in here when I did this. I tried the microphone, and I can't do that. So, I will do better to continuously talk loud. Okay. All right. Going to the liver. All right, the liver, we've talked about it in anatomy. We know what the different lobes are. We know it's separated by different ligaments, right? This is an anatomy. We're just introducing the liver so we can give a framework. All right, and then notice that the gallbladder is also associated with the liver, and that's going to be really important for the function of both the liver and the gallbladder. All right, now the liver has a lot of functions, and depending on what book you look in, the list is going to be like really long or really short. Like, here are the major functions, here are all the functions, okay? I'm not going to be real concerned about a lot of the functions, except what we specifically cover, okay? So, we will be covering formation of bile and some metabolism. You'll see as we go along. All right. You guys already told me that all of the absorption that comes from the stomach, all of the absorption that comes from the small intestine, large intestine, spleen, before it goes to circulation, goes through the liver. And that goes through the hepatic portal vein. All right? Remember, all of those things, veins come from those areas and converge on the hepatic portal vein. That goes directly up into the liver. All right? And if you look, you can't see this very well. That's 75% of the liver circulation. All right? So why do we even have a hepatic artery going to the liver? What's the purpose? What do arteries give that a vein wouldn't? Oxygen, right? So we still need to supply oxygen, and that's where the hepatic artery comes from. All right. So here's how this is organized. Here's all of the veins from different areas. Here's the hepatic portal vein, spreads throughout the entire liver. All right, and as it spreads into the liver, it drains into these hepatic sinusoids that are surrounded, or that surround rather, your liver cells, hepatocytes. And the hepatocytes are the functional cells of your liver. They're the ones that do all the functions that you see on those lists. All right, but what I want you to notice about this is that here's the blood going into the sinusoids. Here are layers of cells that are between the blood and the sinusoids. And within those layers of cells, you have these things called Kupffer cells. And Kupffer cells are like macrophages. They eat things because things you're taking into your gut can potentially get into your blood, and these remove those from the circulation before they head back to the heart. This is one of the reasons why you want blood to go through the liver first. All right, but these other cells that line the sinus, or that line the outside of the hepatocytes, they also provide a filtering function. So only certain things get in, which is most things, but there are certain things you want to keep out. All right, so that's the other function of that. All right, and then everything drains into a central vein, and then that central vein ends up going to the hepatic vein, which then goes to the inferior vena cava, back to the heart, and then back to circulation. All right. Normal liver holds about 10% of our total blood volume. So the liver can act as a blood reservoir. All right, so that if we need extra blood to go into circulation under periods of stress, like exercise, hemorrhage, which is what you see at the bottom of the slide here, then we can draw extra blood to send back to the heart, so we get an increased circulating volume to deal with that stress. Right, more oxygen equals more energy equals more ability to deal with stress. But in conditions such as heart failure, where our heart's really not pumping as much, therefore it can't handle a bigger preload. Ooh, this is where we start entering those words we've already talked about. Bigger preload. The liver can actually increase its holding volume to about 1.5 liters. So you go from about half a liter to holding about 1.5 liters. That's huge. Right? That's a huge reserve volume. And that's all held within these units right here. Okay? These are microscopic, but you have millions of them. It's a large volume. All right. So, the other function of the liver, besides the filtering of blood, which is what we just talked about, and acting as a reservoir of blood, it also forms, stores, and secretes bile. All right? Mostly forming and secreting. Very little is stored because we do have a place to store bile, right? And that's the gallbladder. But it secretes about 0.5 to 1.5 liters a day. That's a lot, right? Bile and certain parts in the bile aid in fat emulsification. Do you remember what that is? You take big drops of fat and convert it into small drops of fat. That's what emulsification does. All right, why do you want to do that? What does that do for the fat? It increases the surface area so that enzymes can break it down easier and then absorb it. Good. All right. Bile is also a medium by which we get rid of bilirubin. All right, bilirubin is a byproduct of red blood cell breakdown. What happens if you have too much bilirubin in your body and in your blood? Jaundice. Good. Let me repeat that, and you're going to thank me for this. When you have too much bilirubin in your blood, what does that lead to? Jaundice. When you go to do your anatomy quiz this week, keep that in mind. Fuckers, don't say I never help you. All right. So, bile is how we get rid of bilirubin and get rid of cholesterol. Yes?
What about bilirubin causes the jaundice itself?
So we get rid of bilirubin. It gets squirted, and we'll talk about this, it gets squirted into your duodenum, and then it doesn't get reabsorbed. It goes all the way through. How do we know we have bilirubin in our digestive tract? It's what gives our poop its color. Okay? What was your question again? I know I answered it, but I don't remember the question.
What makes it actually cause the effect of jaundice?
Okay, so that's what gives poop its color. And if you take poop and wipe it on something white, and I've done this enough as a nurse, wiping people's butts is part of what you do. It's yellow. It's not brown. So that's the color of bilirubin. So if bilirubin's building up in the tissues, it's going to give it that yellow color. That's why your pee's yellow too. Yeah. I got a surprise for you guys in a little bit. Yeah. Not that kind of surprise. All right. All right. So we secrete all this bile, which has a bunch of stuff in it. It travels down this duct system. All right. And in anatomy, we went through what the ducts are. But then some of it gets concentrated into the gallbladder. Is there a question?
No,
Oh.
okay.
Some of it gets concentrated in the gallbladder. So we're secreting all of this. Most of it goes into the gallbladder. The gallbladder is really good at reabsorbing water. And what does that do? That concentrates the bile. All right, so that concentrates the bile so that when it's ready to be secreted, we can secrete concentrated bile into the duct system and into the duodenum to help emulsify fats. All right, so when we make bile, and so go back real quick. Sometimes we get rid of the gallbladder, right? Sometimes we have to take it out because of gallstones, because it's causing other problems. If we get rid of the gallbladder, are we still able to emulsify fats? Yes. Why? We're still secreting it. It's just not being stored. It's just a continuous secretion. Okay? So that's something important to remember. Just because you got rid of your gallbladder, your liver's still making that stuff. It's just not concentrating it and secreting it when needed. It's just continuous secretion. It's also not as concentrated, right? So if it's not as concentrated, does it emulsify as well?
No.
Right. So what would you have to say about your diet then? What would you have to do to your diet? Low fat. Right. All right. Bile secreted in 2 stages. We've seen this picture before. This is just an alternative drawing of it. I did not make it, so I don't draw that well. These hepatocytes, that's what makes the bile. All right. When it makes the bile, it secretes it into these bile canaliculi, these little canals, right, which then go into the bile duct. All right, all empty into the bile duct. Once it's in the bile duct, then the bile ducts, notice that it's lined with cells. Any tube in your body is lined with cells. We know that. That adds water, adds sodium, adds bicarbonate to increase the volume. and allow you to buffer the fluids. All right, that's why the bicarbonate's there, to help you buffer the fluids. Because you have bile acids, you need to buffer that a little bit. All right, so what is in bile? Most abundant is bile salts, right? That's what we're concerned about because that's what provides the main function of bile, are the bile salts. When we talk about emulsifying fats, it's the bile salts that do that, and we'll talk about how that happens. How we form bile salts are made from cholesterol. So when everybody says, "Oh no, you want no cholesterol, don't put any cholesterol in your diet," you actually need cholesterol. Cholesterol has a very important function, not just for bile formation, but also for cell membrane integrity. So keep that in mind. And you get rid of excess cholesterol, and we just through bile. So it gets converted. Cholesterol, fat soluble or water soluble? Fat soluble. But you want it to be water soluble if you want to get rid of it. Bile salts, you want to be partially water soluble. So what you do is you conjugate it. Anytime you see conjugation in relation to constituents of bile, that means adding a hydrophilic amino acid to make it water soluble, or at least one end of it water soluble. Because that's what's flowing, right? You don't have fats flowing through your duct system. You have water flowing through your duct system. So you have to make this stuff water soluble. And so these are just examples of glycine and taurine down here. You don't need to know the structure. I just put it there for completion. All right, so once you do that, that makes bile salts what we call amphipathic. So this is the lipid-soluble portion. This is the water-soluble portion. All right, let's do this. That looks like a sperm, but water-soluble portion, lipid-soluble portion. When it's in the gallbladder, for example, or when it's in the duct system, you have a situation that looks like this. Oops. Right? All the hydrophobic tails are on the inside so that the hydrophilic heads can interact with water. But then once it gets into the duodenum, you have this. Because now it's interacting with fat and separating those molecules. Okay? That's part of the amphipathic nature. Right? And that's how it separates large fat globules into smaller fat globules. All right? By having a hydrophilic end and a hydrophobic end. Does that make sense? Roughly. All right. So, the main constituents, bile salts. When do we need bile salts? When we've had a meal that's high in what? Fat. All right. So when fatty food gets into the duodenum from the stomach, that's going to cause the secretion of CCK, and this is that third function of CCK. CCK is going to go into the blood, go to the gallbladder, and tell the gallbladder to secrete bile. All right, so CCK goes into the blood, goes to the gallbladder, and says secrete bile. So the gallbladder is going to contract. That's going to push bile into the duct system, into where the pancreas also empties into the duodenum, and then allow you to emulsify fats, and then that lipase acts on the fats, as well as those other fat digesting enzymes. All right, does that make sense? So three functions, and Let me see. Yeah, they're all highlighted on your sheet of paper. You're welcome. I don't want to say anything more about that. You get rid of cholesterol through bile too, excess cholesterol. But think about this for a second. Most of your gallstones actually come from concentrated cholesterol. What is your gallbladder good at doing? Concentrating things. So if you have too much cholesterol and you concentrate it too often, you're prone to start creating gallstones. Okay? All right. Back to the small intestine. So secretions of the small intestine, basically we have mucus and a watery fluid. All right, mucus and a watery fluid. That makes sense because we want to protect the lining, we want things to move down smoothly, and we want things to be water soluble. All right. What causes the secretion of these things? Local irritation. That could be anything. Anything going into your small intestine is going to be an irritant to that small intestine. The small intestine is going to respond by secreting more food lube. Right? So things slide down easily. All right. I started asking you questions on Wednesday about where carbohydrate digestion begins, right? Where does it begin? Mouth. With what enzyme? Salivary amylase. Good. All right. Where does carbohydrate digestion finish? Small intestine. Okay. So, amylase breaks down into disaccharides. On your villi and microvilli, you have little enzymes that take disaccharides and break them down into monosaccharides. Okay? And those monosaccharides are then absorbed into the small intestine and into the blood. Make sense? So we've broken it down from you had a potato, a bunch of starch in there. You broke it down into disaccharides, and then those final enzymes on the intestinal wall broke it down into monosaccharides, and you absorb them. We've broken it down to the smallest possible thing we can. Question. Why don't we break down things into their individual atoms, like carbon and hydrogen and nitrogen? You ever thought about that? What?
Not as useful?
They'd still be useful because you can make anything from them, right? They're the building blocks of everything. It'd be very energetically expensive to do that. So you come up with a mechanism where you break them down only so much, and then you can reuse those products. All right, protein digestion. Where does it begin? Stomach. With acid activating pepsinogen into pepsin, right? That starts converting into peptides. Where does protein digestion end? Small intestine with trypsin, chymotrypsin, carboxypeptidase. All right? That's going to break it down from proteins to peptides into individual amino acids, and it's those individual amino acids that are absorbed via active transport. And fats. All right? Fats, their digestion begins where? Small intestine. And it ends in the small intestine. All right. So you're breaking down different types of fats. So it depends on what you're breaking down. Fats, by nature, the lipid membrane is made up of lipids. Fats are lipid soluble. So you don't need active transport. They go right through the membrane. Remember those 8 layers? They travel faster because they go right through membranes. All right? The thing is, though, fat molecules, fatty acids plus monoglycerides, are reformed. They're resynthesized. So you break them down and then you build them right back up, specifically into what the body needs. They package them into these big things called chylomicrons, which is this. So here is a chylomicron with a bunch of phospholipids and proteins. Here's all your fats in the center of it. Notice the hydrophobic tails on the inside with all these triglycerides. They're packaged into this, and then they spit them into the lymphatic system. So they don't go into the blood at all. They get spit right into those lacteals. The lacteals travel along until they get to the cisterna chyli, which is in the abdomen. That goes into the thoracic duct and then empties into circulation. That's what all that says. The only thing that this adds, intestinal villi, remember I said they absorb everything, so that includes water. That just happens by osmosis. Why? You're taking in all these amino acids, all these sugars. They have an osmotic effect. They draw water in. All right, if you want a summary of all of that, I posted a video in your Canvas page that kind of takes you through those steps. All right, now we get to the large intestine, the last part, last 9 slides. Before I talk about the large intestine, though, here's my surprise for you guys. No, I'm not getting undressed. I'm going to poop right in front of you.
Spirit of the wall.
This is one of those white elephant gifts you get. And I wear it to bed. Yeah, and it works. So, now we talk about the large intestine. Otherwise known as the what? Colon. Hopefully this still picks up. All right, large intestine, remember, mostly absorbs water and electrolytes. All right, the water absorption is not just to absorb water. That's what helps actually make our fecal matter. And we'll show how that happens as we go along. So that's one function. The other function is it forms and stores feces. So what do you mean by store? Well, you have a place to put that stuff before it comes out. Okay? All right, and then we've already been through this in anatomy. So the end of the small intestine is the ileum. That goes into the large intestine through the ileocecal sphincter, so that regulates what goes into the large intestine. Then you have this cecum, which is a pouch on the distal end that's attached to the appendix, and that's the start of the large intestine. Then, of course, you have the ascending, transverse, descending, sigmoid, rectum, and anus. All right? Other anatomical features you see, like the flexures, notice that, since this is a physiology lecture, they're not as acutely bent as you see in normal anatomy. But that's the path that they go through. All right? The appendix. One of your cadavers has an enlarged appendix, right? And you saw that. Normally, appendix is very small, about the size of a pinky, maybe even a little bit smaller, and about half as thin. Okay? Does the appendix have a function?
Not that we know.
Not that you know of. Anybody else have an answer?
I think I read something about raw meat digestion, back in the day. It's vestigial from evolutionary stuff.
Okay. And maybe for lower animals that might be true. What they think, and nobody knows for sure, but what they think is let's say you have diarrhea and you've gotten rid of all of your intestinal flora, but there are still some in that appendix. So what the appendix will do is squirt some of that bacteria back into your colon to replenish, a seeding culture for intestinal flora. So that's what they think one of the functions might be. Can you live without it? Yeah, but have you also heard of people who can't do this and have massive flora problems? They give fecal injections into their rectum and anus, and that has been a life-saving procedure. Like, it sounds really gross, but
Don't they have like pills too that you can take because it kind of does like the same thing?
do they? I didn't.
Why don't you just take probiotics instead of milk?
Because so you know all the probiotics you have, and so if you didn't have any,
Yeah, if you're
it's
starting
against them.
Right.
zero, you can't.
There's a difference between prebiotics and probiotics.
All right, so that's we already talked about all of this stuff. So I just wanted to mention something about the appendix. Neat stuff. Here's the absorptive function. All right, when chyme is passing through here, remember you've absorbed a lot of stuff. What is coming here is liquid and left over from what was not absorbed. So that gets squirted into the. I love using words like that because it makes you guys cringe usually. Squirted into the cecum, all right, and then it starts building up, and then you start pushing it this way. This is where you start absorbing water. The purpose of absorbing water is so that as you move along the colon, it gets more solid as you move. So if you were to put a God, what are they called? Colostomy bag into the ascending colon, what would come out is almost liquid. If you were to put it into the transverse colon, it'd be like applesauce-y, maybe a little bit thicker than that. And if you were to put it into the distal colon, descending colon, it's going to be more solid. Okay? Why does that happen? Because as stuff is traveling, you're taking more water out of it, right? So the purpose is not just to get water back into your body. The purpose is to also make a compact form of waste material that you can get rid of. So if you have a condition where you have your colon is too motile, right, moves too fast, what's going to happen to the consistency of your stools? It's going to be non-solid, right? So if you happen to have hyperactive bowels, we talked about listening to bowels in anatomy, all the time, that probably means you have you're pushing things through too fast, and you're likely to have diarrhea. In fact, when you do have diarrhea, you'll hear bowel sounds very hyperactive. Okay, so a couple more terms associated with this. Also, when I sent the announcement out to you guys, I told you, like, when you're bored, laying back there and, like, tracing things. So, like, do that here. So, right side, ascending, transverse, descending. All right. Between the ileum and the cecum, we have the ileocecal sphincter. That is a one-way valve that puts things into the cecum. You don't want things going backwards, right? We talked about, like, the vomiting of fecal matter. That would be really bad. One of the main reasons we already talked about, the colon is full of bacteria. Your small intestines are not really. So if you start having them go backwards, you have a digestive infection. All right. You have these longitudinal smooth muscles, and I hate these drawings because they make it seem like it's only one line going all the way through. There are actually 3 lines of longitudinal muscle going through the colon. Those are called teniae coli. And it turns out that the teniae coli are actually shorter than the colon itself. Okay, and what that produces is a scrunchy effect. So if you have all these longitudinal muscles, the teniae coli, not contracting, they're stretched out like this. Your colon looks really smooth. But when they contract, like a scrunchie, the colon bunches up, and that's what forms those little pouches we call haustra. So when they contract, that bunches up. I like the scrunchie effect. If I had a scrunchie, I'd show you. So what that does is it helps if you have longitudinal contraction, that's moving things in a direction, right? Circular smooth muscle is around the colon. That doesn't help move things along, although it could if you squeeze here, and then squeeze here, and then squeeze here. It's kind of like the cake icing effect. I know that sounds really gross when we're talking about poop. You know, like when you're icing a cake and you push on a bag and it squeezes it out. But mostly it's for mixing things together. All right, and then as we come down, we'll talk about the internal and external anal sphincters the last couple slides. The internal anal sphincter, I will say right now, is under involuntary control. So that takes part in the defecation reflex, the first part. The external anal sphincter is under voluntary control. So when we feel things being pushed along and we go and squeeze, that's the external anal sphincter. Okay, large intestine also secretes a lot of mucus because you have to have a medium to push this stuff through, especially as it gets more solid. And the mechanism by which the large intestine absorbs water, and the reason why it absorbs water and electrolytes, you absorb a lot of sodium, absorb a lot of chloride. What's going to come with it? Water by osmosis. So it's not a direct, like, hey, bucket brigade into the venous system. Water's following electrolytes. All right. Why do we have bacteria? We talked about bacteria. Why do we even have it in our large intestine? What's the point? What does it do for us? Well, we eat a lot of things that we can't break down. Our body simply can't do it. So these bacteria in the large intestine do help break down those things like cellulose. I mean, we don't have enough bacteria in our gut, and corn is not in our large intestine for a long enough period of time for all of that bacteria to break down that cellulose. That's why corn appears in our fecal matter without being fully digested. But that it is still made out of cellulose. All right, they also synthesize some vitamins, right? So some vitamins that we need, bacteria synthesize them. They get absorbed and we benefit, right? But they also produce gas, right? Which is why you produce flatulence that smells. All right, as far as the movements go, of course, we talked about the mixing movement. Those would be the circular muscles. The tenia coli are what produce the peristaltic waves. And usually when we have massive peristaltic waves, they occur in about, 2 to 5 mass movements. All right? That's what moves things down, and that's what pushes stuff into our rectum and stretches it and starts the defecation reflex. Okay, so these mass movements push into the rectum, and that's what starts the defecation reflex. So that's the last thing we're going to talk about. Fecal matter specifically, what's in it? Water, electrolytes that weren't stuff that wasn't absorbed, mucus because that's what helped it get to where it is, bacteria because that's what's in our large intestine, bile pigments, bile salts, cholesterol, that's what gives it its color. All right. Several years ago, Burger King came out with this gimmick where they had the Halloween hamburger. You guys remember this?
Oh, the black one.
And it was a black bun. And I was like, ooh, that looks neat. I'll try it. For the next 2 days, This was the color of my fecal matter.
Oh gosh.
Like it was a bright, actually no, it was more like that water bottle right there. It was a bright green because of the dyes that never got absorbed that ended up in fecal matter. So you can change the color of your fecal matter by what you eat.
That's how they track animal poop in the zoo. They give them glitter so they can know what it looks like.
Yeah. But also, you can tell if there are problems, especially with the liver, by the color of fecal matter too. If it is very gray, then that means bile isn't getting into your colon. That means bile isn't getting into your small intestine, and you may have a problem with your gallbladder, the duct system, your liver. All right, so the color of poop's very important, like very good diagnostic tool. Can tell you what's going on in your digestive system. All right, so lastly, the defecation reflex. There are two parts to this. One is an intrinsic defecation reflex. When I say intrinsic defecation reflex, where is the signal for that coming from? The enteric nervous system. All right, so mediated entirely by the ENS. That's the enteric nervous system. All right, when feces, when fecal matter enters the rectum, all right, there are stretch receptors there that stretch, send signals to the enteric nervous system that starts causing contractions and moving whatever's in the large intestine along, all right, to help you get rid of whatever is stretching the rectum. All right? When that happens, the internal anal sphincter relaxes and fecal matter starts coming out. All right? That happens automatically. When children are potty training, what they're learning to do is control that external anal sphincter to clamp it closed despite the internal anal sphincter being relaxed and open. Okay? So by that mechanism and the external anal sphincter, we can voluntarily control defecation. Okay? Now, we also have a defecation reflex that acts through the spinal cord. So that stretching will send signals to the spinal cord in the sacral area. The spinal cord will then send signals back down to the rest of the large intestine and signal peristaltic waves. What that does is it just makes the contraction stronger. All right? What it's doing specifically is, like it says there, qualitatively the same thing. You're still giving nerve signals to cause contractions to push poop. All right? Firstly, it comes from the enteric nervous system. The spinal cord reflex intensifies that. Okay? All right, but now